Hello, I'm Jasmine Trigg, a scientist at SARTORIUS. Today I'll be discussing a poster previously presented at the American Association of Immunologists conference that was titled Multiparametric Quantification of Monocytes using Live Cell Analysis. Monocytes play critical roles in immunity by migrating to inflamed tissue, where they clear microorganisms and apoptotic cells, repair injured tissues, and recruit other immune cells. These highly plastic cells can change their functional phenotypes in response to a variety of cellular signals, including the AKT signaling pathway. Here we provide robust in vitro assays for the kinetic evaluation of monocytes and exemplify how these further our understanding of their biological role. The InKsight live cell analysis system was used to acquire phase and fluorescence images of monocytes, and these were automatically analysed using integrated software. If we look at the panel at the top, this shows the InKsight live cell analysis system, which is a fully automated phase contrast and multicolour fluorescence system that resides inside a standard cell culture incubator for optimal cell viability. It's designed to scan plates and flasks repeatedly over time. In combination with our software, this offers fast, flexible and powerful control for continuous live cell analysis. We also offer a suite of consumables and non-perturbing cell labelling and reporter reagents. Additionally, cytokine analysis was performed using the IQ Advanced Flow Cytometry platform and a custom QB Plex screen kit. These data exemplify that live cell analysis, alongside advanced analytical methods, is a powerful approach enabling multiparametric quantification of immune cells. The activation and differentiation of immune cells is often accompanied by morphological changes, and we can quantify these using the InKsight Advanced Label-Free Classification software module. This software enables automated quantification of cell morphology by employing multivariate analysis to identify multiple morphological features, such as cell area, texture, brightness, and symmetry. These parameters are then used to create an unbiased, meaningful score value that enables subpopulations to be classified into two user-identified groups. Here we performed a label-free differentiation assay using THP1 monocytes, and these were differentiated to a macrophage phenotype using PMA in the presence of Fab448A labelled CD11B, and this is a macrophage marker. Images were acquired over 3 days and segmented using the InKsight cell-by-cell analysis software module. Macrophages were quantified using both advanced label-free classification and fluorescence classification of the CD11B marker. The phase and fluorescence blended images are shown here, and these show the change in morphology and increase in CD11B expression as the cells differentiate from a monocyte to macrophage phenotype. Also shown are the label-free classification masks, with the monocytes segmented in pink and the macrophages with the altered morphology shown in blue. The time course of the percentage of macrophages shown here shows that accompanying differentiation, we have an increase using the label-free classification that is comparable to what we see for the increase in CD11B expression. Following the differentiation of monocytes, we can then perform functional characterization to assess the biological significance. In this example, we're assessing cell chemotaxis, an essential component for understanding the immune response. THP1 cells, either naive or PMA differentiated, were seeded onto InKsight Clearview 96-well plates, and these were coated with fibronectin. The cells were then exposed to the chemoattractant C5a. Integrated InKsight chemotaxis analysis software was used for the real-time quantification of the cells as they moved through the pores and adhered to the bottom side of the membrane. If we look at their time course and transform data, we can see that the differentiated macrophage, like THP1 cells, had a concentration-dependent chemotactic response towards C5a, whilst little to no response was observed in naive THP1 cells. The results reveal the functional significance of monocyte maturation in chemotactic migratory responses. Monocytes are highly plastic cells, and they can change their functional phenotypes in response to a variety of cellular signals, including the AKT signaling pathway. To monitor AKT activity, we use cells that are expressed in the incucyte kinase AKT green-red biosensor. And this is a genetically encoded fluorescence reporter whose subcellular localization is dependent on AKT activation. Raw monocytes expressed in the biosensor were serum starved for 3 hours to initially inactivate AKT, and then they were treated with a concentration range of lipopolysaccharide, or LPS. If we look at the green fluorescence images, these show that there's translocation from the nucleus to the cytoplasm for LPS-treated monocytes compared to vehicle. When we look at the quantification, we observe that LPS induced a rapid concentration-dependent increase in the nuclear translocation ratio, and this indicates AKT activation. We also performed cytokine analysis using the IQ advanced flow cytometer and a custom Q-beads plex screen kit. LPS stimulated release of the inflammatory proteins TNF-alpha and CCL3, consistent with LPS inducing activation by the release of pro-inflammatory reagents. Additionally, we performed a label-free morphological assessment of the effect of immune cell activation in response to LPS. Raw monocytes were stimulated with increasing concentrations of LPS, and the phenotypic morphological changes over 2 days were quantified using the incucyte advanced label-free classification analysis. The time course shows LPS induced a concentration-dependent increase in the percentage of activated cells as cells become adherent, larger and flatter. We observed a maximal response of around 56% for the 100 nanogram per ml LPS at 33 hours. Representative images are also shown of the observed rapid changes in morphology following LPS stimulation. Also shown in the classification mask for the monocytes in pink and the activated macrophage-like cells shown in blue over 30 hours. Another critical role monocytes play in innate immunity is the clearance of microorganisms and apoptotic cells. Here we use the incucyte phagocytosis assay and AI-driven analysis to assess the phagocytic capability of monocytes. The incucyte phagocytosis assay utilizes Frodo reagents for incucyte, and these use a pH-sensitive conjugated fluorophore that, following engulfment and phagolysosome formation, increase in fluorescence intensity due to an increase in the acidic environment. This increase in fluorescence can then be automatically quantified over time using integrated software. In this study, raw monocytes were treated with Frodo E. coli bioparticles for incucyte and analysed using the incucyte AI cell health analysis. So we've got some representative images shown here on the left of monocytes. We can see that the non-engulfed bioparticles are visible in the phase image, whilst the engulfed bioparticles fluoresce green. The AI cell segmentation mask is shown in yellow, and you can see how this allows for accurate masking of cells and is capable of ignoring the bioparticles in the background. The microplate graph reports the increasing green fluorescence intensity within the segmented cell boundary over 24 hours and highlights the assay robustness. We observed a kinetic concentration-dependent increase in the cellular fluorescence as the bioparticles were engulfed by the monocytes. In addition to the clearance of microorganisms and apoptotic cells, the uptake and clearance of viable tumour cells can be promoted with monoclonal antibodies. This can occur via antibody-dependent cellular phagocytosis, also known as ADCP, or through the blockade of don't eat me signals such as CD47. These mechanisms hold immunotherapeutic promise and are studied extensively in drug development. Here we investigated how anti-CD20 monoclonal antibodies promote ADCP in macrophages using the incucyte phagocytosis assay. Primary monocytes were isolated from whole blood and differentiated into macrophages using cytokines. Frodo for incucyte labelled Ramos cells were treated with monoclonal antibodies targeting anti-CD20, and these were rituximab or its biosimilar Truxima, or IgG isotype control. and these are then co-cultured with macrophages. Macrophage differentiation was monitored and quantified using the INKSIGHT Advanced Label-Free classification analysis. And you can see the images here that show classification masks for the monocytes in pink and for the macrophages in blue on day 0 and day 7 respectively. If we look at the time courses on the right here, these show that rituximab and truximab induce ADCP in a concentration-dependent manner, with similar kinetic profiles being observed over the 4-hour time period. So to conclude, the INKSIGHT live cell analysis system and integrated software provides an image-based solution for the kinetic evaluation of monocytes in real time. Our experimental findings demonstrate the quantification of the changes accompanying monocyte differentiation and activation. Additionally, we exemplified the functional characterization of immune cells using measurements of AKT activity, chemotaxis, and phagocytosis. These data exemplify that live cell analysis, alongside advanced analytical methods, is a powerful approach enabling multi-parametric quantification of immune cells. Thank you for listening to this presentation.